Hi everyone. Welcome. If you're here right now, you're probably getting ready to rest. Maybe you've already turned the lights down low. Maybe you're lying in bed or settling into a comfortable chair. Whatever you're doing, you're welcome here. This video is going to move slowly. It's going to be quiet, and it's going to take a very long, very gentle look at one of the most ordinary things in the natural world. We're going to talk about leaves. More specifically, we're going to talk about why leaves are green. Now, you might think that's a simple question. You might think the answer will take only a moment, but it turns out that the greenness of a leaf is one of the most quietly extraordinary things in all of biology. It touches on light and chemistry and the deep history of life on Earth. It connects to stars and to the ocean and to the very air that you're breathing right now. But we're going to take our time getting there. There's no hurry tonight. We'll move slowly through the ideas, the way you might wander through a quiet forest in the late afternoon. No rushing, no urgency, just a gentle walk through one of nature's most beautiful secrets. So let's begin. Let's start with something very simple. Let's start with a leaf. Think about a leaf. Any leaf will do. Maybe it's a large, flat maple leaf, the kind that falls in autumn. Maybe it's a long, narrow blade of grass. Maybe it's the small, rounded leaf of a houseplant sitting on a windowsill. It doesn't really matter which leaf you imagine. Almost all of them share something in common. They're green, not just a little green. Most leaves are deeply, richly, persistently green. During the spring and summer months, the world fills up with this color. Forests become walls of green. Roadsides blur with it. Gardens overflow with every shade. Pale lime, deep forest green, yellow-green, blue-green, and every variation in between. But underneath all of those shades, most leaves share the same basic green pigment. And the reason they have that pigment, the reason they are green at all, is one of the most elegant stories in all of science. Let's start at the very beginning. To understand why leaves are green, we first need to understand what a leaf is actually trying to do. Because the color isn't just decoration. It isn't random. It is a direct result of the leaf's most important job. And that job is to capture light from the sun. A leaf is, at its heart, a solar panel. Not a mechanical one, of course. Not made of silicon or wires. But a leaf does something very similar to what a solar panel does. It absorbs energy from sunlight and uses that energy to do work. The work that a leaf does is called photosynthesis. Photosynthesis is the process by which a plant takes sunlight, water, and carbon dioxide, a gas from the air, and turns them into sugar. That sugar is food. It's the energy source that powers the entire plant. It powers the growth of stems, and roots, and flowers, and fruit. It powers everything. And photosynthesis happens inside the leaf. Now, when we say photosynthesis happens inside the leaf, we mean something very specific. We mean it happens inside tiny structures within the leaf cells. These structures are called chloroplasts. Chloroplasts are small, very small. You cannot see them without a microscope. Each one is smaller than a grain of sand by many thousands of times. And yet, inside each chloroplast, something remarkable is constantly happening. Light energy from the sun is being captured, transformed, and stored. It's happening right now. In every leaf you can see, quietly, continuously, without any fuss at all. And inside those chloroplasts, there is a molecule, a single type of molecule, that is responsible for the greenness of nearly all plants on Earth. That molecule is called chlorophyll. Chlorophyll. It's worth saying the word slowly. Chlor, O-P-H-Y-L-L. It comes from the Greek words for green, chloros, and leaf, phyllan. So chlorophyll simply means, in a literal sense, green leaf. The molecule is named for the color it gives. Chlorophyll is a pigment. A pigment is any substance that absorbs certain wavelengths of light and reflects others. When light hits a pigment, some of that light is absorbed, and some of it bounces back. The color we see is the color of the light that bounces back, the light that wasn't absorbed. And chlorophyll, when light hits it, a absorbs most of the red and blue light very strongly. It does not absorb green light very well, so the green light bounces back. And that's what our eyes see. That's the green of the leaf, not the light the leaf is using, just the leftover light, the light that didn't get absorbed, the light that was reflected back into the world. There's something almost meditative about that. If you think about it gently, the green color of a leaf is what the leaf didn't need. It's the remainder, the leftover, the color that the plant couldn't use, was sent back out into the world, and it became the most abundant color in the living landscape. But why does chlorophyll absorb red and blue, and not green? That's the deeper question. And to answer it, we need to go back even further. We need to talk about light itself. Light is a kind of energy. You already know that. But what light actually is, at a physical level, is worth taking a moment to consider. Light travels in waves, like ripples on water, but much, much smaller, and moving much, much faster. Light travels at about 300,000 kilometers per second. That's fast enough to circle the entire Earth, more than 7 times in a single second. Now, different colors of light have different wavelengths. A wavelength is simply the distance between 2 peaks of a wave, the distance from one ripple crest to the next. Red light has a relatively long wavelength. Blue light has a shorter wavelength. Green light falls somewhere in between. When all of these wavelengths are mixed together, we see white light. Sunlight, which contains all of these wavelengths, looks white or pale yellow to our eyes. But when light passes through a prism, a triangular piece of glass, the different wavelengths bend by different amounts. They separate, and we see the full spectrum: red, orange, yellow, green, blue, indigo, violet. A rainbow. That rainbow is always there, hidden inside sunlight. It just takes something to pull it apart, so we can see it. And a leaf does something like the opposite of that. A leaf takes the full spectrum of sunlight and absorbs most of it. It selectively removes the red and the blue and sends back the green. The result is the green world we live in. Now, let's look at chlorophyll a little more closely. Chlorophyll is a molecule. A molecule is a group of atoms bonded together, and chlorophyll is a fairly large molecule. By the standards of chemistry, it has a specific, carefully arranged structure that makes it particularly good at absorbing light. The most important part of the chlorophyll molecule is a structure called a porphyrin ring. A porphyrin ring is a flat, roughly circular arrangement of atoms. It's made mostly of carbon and nitrogen, arranged in a ring-like pattern, and at the very center of that ring sits a single atom, a magnesium atom, just one, right in the center. That magnesium atom is crucial. It's what makes the chlorophyll molecule behave the way it does. It's what allows the molecule to interact with light in such a specific, precise way. Attached to the porphyrin ring is a long tail, a chain of carbon and hydrogen atoms that makes the molecule somewhat like a flat head, with a long ribbon attached. This tail is called a phytol tail, and it helps anchor the chlorophyll molecule inside the chloroplast, keeping it in the right position to do its job. The whole molecule is a beautiful piece of natural engineering, flat enough to absorb light efficiently, anchored carefully in place, centered on a single metal atom. That makes everything work. And there are actually 2 main types of chlorophyll. In most plants, chlorophylla and chlorophyll B. They're very similar to each other. The main difference is a small chemical group attached to one part of the porphyrin ring. Chlorophylla absorbs light most strongly in the blue-violet region and in the red region. Chlorophyll B absorbs light slightly differently. It prefers a slightly different shade of blue and a slightly different shade of red. Together, they cover a broad range of the spectrum. Together, they make the leaf more efficient at capturing light across a wide range of conditions. It's a kind of partnership, 2 very similar molecules working together, covering slightly different territories. So how exactly does a molecule absorb light? To understand this, we need to think about electrons. Every atom has electrons. Electrons are tiny negatively charged particles. that orbit around the nucleus of an atom. And electrons exist in specific energy states. They can be at a lower energy level or a higher energy level, but they can't be at any energy level. They can only occupy certain specific levels, like steps on a staircase. When a photon of light, a tiny packet of light energy, hits an electron, something can happen. If the photon has exactly the right amount of energy to push the electron up to a higher step, on that staircase, the electron absorbs the photon. The electron jumps up. Different molecules have different staircases, different gaps between the steps, and only photons with exactly the right energy, which means exactly the right wavelength, exactly the right color of light, can make that jump happen. Chlorophyll's molecular structure happens to have energy steps that match up with red light and blue light. When a photon of red light hits a chlorophyll molecule, its energy exactly matches the gap needed to push an electron upward. The electron absorbs that energy. The photon disappears. The electron is now in an excited state. The same thing happens with blue light. But green light, the energy of green light photons, doesn't quite match any of the energy gaps in chlorophyll. So green photons aren't absorbed. They're reflected or transmitted. They pass on through, and we see green. It's a very precise, very specific thing. Not a choice the plant makes. Not something the plant controls in a conscious way. It's just the natural consequence of the shape and structure of a molecule interacting with the physics of light. The greenness of leaves is written into the laws of physics and chemistry. So an electron in a chlorophyll molecule has absorbed a photon. It's been pushed up to a higher energy state. Now what? This is where photosynthesis really begins. The excited electron, now carrying extra energy, is passed along a chain of molecules inside the chloroplast. This chain is called the electron transport chain. It's a series of carefully arranged proteins and molecules that pass the electron from one to another, like a relay race. As the electron moves along the chain, it gives up its energy in small, controlled steps. That energy is used to do chemical work, specifically to build a molecule called ADP. ADP stands for adenosine triphosphate. It's the universal energy currency of life. Almost every living thing on Earth uses ADP to power its chemical reactions. When a cell needs to do something, move, grow, repair itself, it uses ADP as the fuel, and photosynthesis makes ADP from sunlight. But that's not all. Photosynthesis also does something else. With the energy from light, it uses that energy to split water molecules. Water, as you know, is H2O, 2 hydrogen atoms and 1 oxygen atom. Inside the chloroplast, water molecules are split apart. The oxygen is released. It drifts out of the leaf and into the air. That oxygen is what we breathe. The oxygen in every breath you take right now came ultimately from this process, from water molecules being split inside chloroplasts, inside leaves, all over the world. Think about that gently for a moment. The oxygen that is moving through your lungs right now, that is sustaining your life, as you listen to this, was released by leaves, by the splitting of water molecules inside tiny chloroplasts, somewhere out there in the plant world. It's a quiet, continuous gift. Plants don't even notice they're doing it. After the light-driven reactions produce energy and split water, there is another stage of photosynthesis. This stage is called the Calvin cycle. It's named after Melvin Calvin, an American chemist who figured out the details of this process. In the 1950s, he received the Nobel Prize in Chemistry. In 1961, for this work, the Calvin cycle is sometimes called the dark reactions of photosynthesis, not because they only happen in the dark, but because they don't directly require light. They use the energy stored in ADP and other energy-carrying molecules to do their work, and their work is to take carbon dioxide from the air and build sugar molecules from it. Carbon dioxide is a simple molecule. One carbon atom bonded to two oxygen atoms. CO2. It's the gas that you breathe out. It's also present in the air. At low concentrations, about 400 parts per million, leaves absorb carbon dioxide through tiny pores in their surface, called stomata. We'll talk more about stomata later. For now, the important thing is that carbon dioxide enters the leaf, drifts into the cells, enters the chloroplasts, and becomes part of the Calvin cycle. In the Calvin cycle, carbon dioxide molecules are attached to a larger molecule, one at a time, building up a chain of carbon atoms. With the energy from ADP and related molecules, these carbon chains are rearranged and reshaped until they form glucose, a simple sugar. Glucose is the fundamental food molecule of the plant. It can be used immediately for energy, or stored as starch, or converted into all the other molecules the plant needs. The cellulose of cell walls, the proteins, and fats, and pigments that make up the plant's body. Everything the plant is made of, ultimately, comes from carbon dioxide, from the air. That's a remarkable thing to sit with. A tree that weighs thousands of kilograms, a great oak or a redwood, built nearly its entire body from air, from carbon dioxide, pulled one molecule at a time, from the atmosphere, combined and rearranged by sunlight and chlorophyll. Most of a tree's mass came from the air, from the same air you're breathing right now. Let's just pause here for a moment. We've been moving through some chemistry and some physics, and it's all very gentle, but it can feel like a lot. So let's just settle a little. You're doing well. You don't need to hold all of this tightly. Let the ideas drift through you, like the light drifting through a leaf. You don't need to remember every detail. You don't need to understand every step perfectly. Just let it wash over you. The sun shines. The leaf absorbs. Electrons jump. Water splits. Sugar forms. Oxygen flows. This is happening right now. In every leaf in the world that has light on it. In the trees outside. In houseplants on windowsills. In algae in ponds. In mosses on rocks. In the tiniest blades of grass, pressing up through sidewalk cracks. The whole green world is busy quietly, making sugar from sunlight. And all of that incredible machinery, is because of chlorophyll, because of the specific way that molecule absorbs red and blue light, but not green. Let's go back into the chloroplast. Let's look at its structure a little more closely. A chloroplast is a small, oval-shaped structure inside a plant cell. It's surrounded by two membranes, two thin layers that act like walls, keeping the inside separate from the outside. Inside the chloroplast, there's a fluid called the stroma. The Calvin cycle happens here. In the stroma, it's the more liquid, open part of the chloroplast. But inside the stroma, there are also stacks of membranes. These stacks are called grana. The singular is granum. Each granum is a stack of flattened disc-like structures called thylakoids. Thylakoids are the membranes where the light reactions happen. The chlorophyll molecules are embedded in thylakoid membranes, arranged in clusters, called photosystems. When light hits a photosystem, the chlorophyll molecules work together to capture that energy and begin the electron transport process. The arrangement is very orderly, very deliberate. The thylakoid membranes are stacked up so that they have a large surface area inside a small space. More surface area means more room for chlorophyll molecules. More chlorophyll molecules means more light can be captured. It's an efficient design, compact and well organized, like a library where the books are carefully shelved to fit as many as possible into a limited space. There are typically somewhere between 10 and 100 chloroplasts in a single leaf cell, and a single leaf contains millions and millions of cells, so the total number of chloroplasts in even a small leaf is enormous, and in each of those chloroplasts, thousands of light reactions are happening every second. The leaf is always busy, even when it looks perfectly still. Now let's talk about something you might have noticed in real life. Leaves aren't all the same shade of green. Some leaves are a deep dark green, the kind you see on holly bushes or on ivy growing up old walls. Some leaves are a bright, fresh, almost yellow-green, like the new leaves of spring or the soft fronds of ferns. Some leaves have a blue-green tint, like eucalyptus. Some are a pale, washed-out green, almost silvery. What causes these different shades? Several things. First, the concentration of chlorophyll matters. Leaves with more chlorophyll tend to be darker green. Leaves with less chlorophyll are lighter. The total amount of the pigment, how densely it's packed into the leaf, determines how saturated the green color appears. Second, there are other pigments in leaves besides chlorophyll. Carotenoids, for example. These are yellow and orange pigments that are present in most leaves, alongside chlorophyll. Usually, the chlorophyll is so dominant that it hides the carotenoids completely. We just see green, but the yellow is always there, underneath, waiting. In autumn, when chlorophyll breaks down, the yellow and orange carotenoids are revealed. The hidden colors emerge, but we'll come back to autumn in a little while. Third, the structure of the leaf itself affects how green it looks. Some leaves are thick and waxy, with a reflective surface. They reflect more light before it can even enter the leaf. These tend to look a shinier, brighter green. Other leaves are thin and somewhat translucent. They allow more light to pass through, and they look softer, more delicate. And then there is light itself, and the way we perceive color under different conditions. A leaf in bright midday sun looks different from the same leaf in the soft light of early morning. The green shifts and changes throughout the day. Green is not one color. It is a whole family of colors, varying with chemistry and structure and light. Let's take a moment to look at the structure of a leaf itself, not just the chemistry inside, but the physical architecture. A typical leaf has several layers. On the outside, top and bottom, is the epidermis. This is a thin layer of cells that acts like a skin for the leaf. The top epidermis is often covered with a waxy layer called the cuticle. The cuticle is waterproof. It helps the leaf retain moisture and protects it from drying out. In the sun and wind, just below the upper epidermis, is a layer called the palisade mesophyll. This is where most of the photosynthesis happens. The cells in this layer are tall and tightly packed, like columns standing upright. They're densely filled with chloroplasts, arranged to catch as much of the incoming sunlight as possible. Below the palisade layer is the spongy mesophyll. The cells here are more loosely arranged, with air spaces between them. These air spaces are important. They allow carbon dioxide to diffuse through the interior of the leaf, reaching all the cells that need it for photosynthesis. Running through all of these layers are veins. Leaf veins are bundles of two types of tissue, xylem and phloem. Xylem carries water from the roots up into the leaf. Water is essential for photosynthesis. It's one of the raw materials that gets split apart in the light reactions, releasing oxygen. Phloem carries sugar out of the leaf after photosynthesis has made glucose. That glucose needs to be distributed to the rest of the plant, to the growing tips of stems, to the roots, to the developing fruits. The phloem is the transport system that makes this possible. The veins of a leaf, when you look at them against the light, are beautiful. They branch and branch and branch again, reaching every part of the leaf with water and carrying sugar away. No part of the leaf is more than a tiny distance from a vein. Let's talk about stomata. Stomata are tiny pores, tiny openings in the surface of a leaf. The word stomata comes from the Greek word for mouth, and they are, in a sense, the mouths of the leaf. Through stomata, the leaf breathes. Carbon dioxide comes in through stomata, oxygen goes out through stomata, and water vapor also escapes. through stomata. This is called transpiration, and we'll get to that in a little while. Most stomata are on the underside of the leaf, away from direct sunlight. This helps reduce water loss. If the stomata were on the top, facing the sun, they would dry out more quickly. Each stoma, that's the singular form of stomata, is surrounded by two special cells, called guard cells. Guard cells are shaped a bit like sausages, or like the two halves of a pea pod. They're hinged at each end. When the guard cells swell with water, when they absorb water and inflate like tiny balloons, they curve outward, and as they curve outward, a gap opens between them. The stoma opens when the guard cells lose water. When they deflate, they straighten out again, and the gap closes, so the plant can control whether its stomata are open or closed. It can open them to let in carbon dioxide when it needs to photosynthesize. It can close them to conserve water when conditions are dry. This is a very elegant system. No brain required, no conscious decision, just a physical mechanism, the inflation and deflation of two curved cells that opens and closes, a tiny pore. In response to conditions, light signals the guard cells to open. The leaf opens its stomata. In daylight, ready to photosynthesize. In darkness, the stomata tend to close. There's no photosynthesis happening, no need for carbon dioxide, and no reason to risk losing water. A single leaf may have hundreds of thousands of stomata arranged on its underside, like a field of tiny invisible mouths, each one opening and closing throughout the day in response to light, temperature, humidity, and the plant's water status, all quietly, all continuously. Let's think about water for a moment. Water is one of the most important substances in the life of a leaf. It's a raw material for photosynthesis. It provides structure. When leaf cells are full of water, they're firm and stiff, and the leaf holds its shape. When a plant loses too much water, the cells deflate slightly, and the leaf droops. That drooping, wilting, is the plant's way of showing that it needs water. Water enters the plant through the roots, root hairs, tiny delicate extensions of root cells absorb water from the soil. That water moves upward through the plant. In the xylem, the water-carrying tubes that run through stems and branches and into leaves. How does water get from the roots all the way up to the top of a tall tree, even against gravity? This is one of the more wonderful puzzles in plant biology. The answer involves transpiration, the evaporation of water from the stomata of leaves. When water evaporates from the surface of a leaf cell and exits through a stoma, it creates a slight tension, a pulling force, in the water column inside the leaf. This tension is transmitted down through the veins, through the stem, all the way down to the roots. It's like drinking through a straw. When you sip, you create suction. That suction pulls liquid up through the straw. Transpiration does the same thing. The evaporation at the top pulls water all the way up from the roots. The scientific name for this is the cohesion tension theory. Cohesion refers to the tendency of water molecules to stick together. Water is quite sticky at the molecular level, so the whole water column pulls as one connected piece. Tension refers to the pulling force created by evaporation. At the top, trees can be very tall. A mature coast redwood can be over 100 meters tall. Water must travel from roots in the ground all the way to leaves. At the very top, over 100 meters of vertical distance. And it does this not by being pumped exactly, but by being pulled upward by the gentle, continuous evaporation of water from millions of leaves, quietly, continuously, one molecule at a time. Let's just let that sit for a moment. We've covered a lot of gentle ground. Light and electrons, chlorophyll and color, the structure of the leaf, the tiny mouths in the leaf skin, water moving upward through trees against gravity. All of this is happening in the leaves outside your window, in the trees along your street, in the parks and gardens and forests of the world. Everything is very busy and very quiet at the same time. You don't need to hold any of this tightly. Just let it drift. Maybe your eyes are getting a little heavier now. Maybe your breathing has slowed down. Just a little. That's good. That's exactly right. We'll keep going, nice and gently, through more of the story of why leaves are green. There's quite a lot more to explore. We have all the time in the world. Now let's step back in time. A long way back. The Earth is about 4.5 billion years old. And for the first 2 billion years or so of its history, there was very little oxygen in the atmosphere. The early Earth's atmosphere was mostly made up of nitrogen, carbon dioxide, methane, and water vapor. There were no plants. There were no animals. There were no leaves. But there was life. The earliest life on Earth was microbial, single-celled organisms, invisible to the naked eye. These early microbes lived in the oceans, and they had various ways of getting energy. Some 8 organic compounds that formed from chemical reactions. Some got energy from chemical processes in rocks. And then, somewhere around 2.5 to 3 billion years ago, something new appeared. A type of microbe that could capture energy from sunlight. These were the first photosynthetic organisms. They were called cyanobacteria, and they changed everything. Cyanobacteria are still alive today. They live in oceans, lakes, rivers, and soils all over the world. They're sometimes called blue-green algae, though they're not technically algae. They're bacteria, and they photosynthesize. The cyanobacteria of billions of years ago slowly released oxygen into the atmosphere as a byproduct of their photosynthesis. At first, this oxygen was absorbed by iron in the oceans. It reacted with dissolved iron to form iron oxide, which settled to the sea floor. Ancient rocks, called banded iron formations, record this process. They're striped with layers of rust-colored iron oxide, laid down over hundreds of millions of years. But eventually, so much oxygen had been produced that it began to accumulate in the atmosphere. This was called the Great Oxygenation Event. It happened roughly 2.4 billion years ago, and it was, in a sense, the most dramatic change in Earth's atmosphere in its entire history. For many microbes at the time, oxygen was actually toxic. It was a dangerous reactive gas that damaged their cells. The Great Oxygenation Event likely caused a massive die-off of anaerobic microbes, organisms that had evolved in an oxygen-free world. But for other organisms, oxygen was an opportunity. Oxygen can be used for a much more efficient kind of energy production, aerobic respiration. Organisms that could use oxygen to break down food molecules could get far more energy from the same amount of food. And so, the history of complex life on Earth was set in motion by cyanobacteria, by photosynthesis, by the slow accumulation of oxygen in the air. Now here's a fascinating idea, a gentle but radical one. Where did the chloroplasts in plant cells come from? The answer takes us back to those ancient cyanobacteria. About 1.5 to 2 billion years ago, something remarkable is thought to have happened. A large single-celled organism, a kind of early eukaryote, a cell with a nucleus, engulfed a cyanobacterium. Normally, when a cell engulfs something, it digests it, it breaks it down for food. But this time, something different happened. The cyanobacterium wasn't digested. Instead, it survived inside the larger cell, and the two organisms formed a kind of partnership. The cyanobacterium continued to photosynthesize inside the larger cell. The larger cell provided the cyanobacterium with protection and raw materials. In exchange, the cyanobacterium provided photosynthetically made sugars to the larger cell. Over time, the cyanobacterium became less and less independent. It transferred many of its genes to the nucleus of the host cell. It became permanently incorporated. It became, in essence, an organelle. That organelle is the chloroplast. This idea is called the endosymbiotic theory. Endo means within. Symbiotic means living together in a mutually beneficial relationship. The theory says that chloroplasts originated as free-living cyanobacteria that were engulfed by larger cells and became internal partners. The theory was proposed and developed primarily by a biologist named Lynn Margulis in the 1960s. At first, it was not well received by the scientific community. Many scientists found it strange, even far-fetched, but the evidence accumulated over the years. And today, the endosymbiotic theory is one of the most well-supported ideas in all of biology. The evidence is compelling and rather beautiful. Chloroplasts have their own DNA, not the same DNA as the rest of the cell, their own separate, circular DNA, very similar to bacterial DNA. This is exactly what you'd expect. If chloroplasts descended from a bacterium, chloroplasts reproduce by dividing in two, just as bacteria do, not in the same way that other organelles form. The double membrane surrounding a chloroplast, two layers, not one, is consistent with the idea that the inner membrane was originally the membrane of the engulfed bacterium, while the outer membrane came from the host cell's engulfing process. And if you look at the molecular structure of chloroplasts, the proteins they're made of, the genes that encode those proteins, they are clearly related to cyanobacteria. The chloroplast in the leaf of a plant is a descendant of a free-living ocean bacterium that was swallowed whole 2 billion years ago and never left. That's a quiet, remarkable thing. The green in every leaf is ancient. It carries within it the echo of a much older world. This brings up an interesting question that we should address gently. If plants need light so badly, if capturing sunlight is the whole point of the leaf, why do they reflect green light? Why not absorb all the light? Wouldn't a perfectly black leaf be better at photosynthesis, since black absorbs all wavelengths? This is a question that scientists have thought about quite a bit, and the answer is, as with most things in biology, a bit complicated. One part of the answer involves the history of evolution. Chlorophyll absorbs red and blue light efficiently. It does this because of its particular molecular structure, the structure that evolved in cyanobacteria billions of years ago, long before there were any plants. The specific arrangement of atoms in the porphyrin ring, with magnesium at the center, happens to absorb those particular wavelengths. Natural selection didn't design chlorophyll from scratch, optimizing it for maximum light absorption. It modified what already existed. The chlorophyll that we see in plants today is descended from the chlorophyll of those ancient cyanobacteria. It was good enough to allow photosynthesis to thrive, so it was kept. Not perfect, just adequate. Better than nothing. Evolution doesn't produce perfect solutions. It produces solutions that are good enough. But there's another interesting idea worth mentioning. Some researchers have proposed that green light might actually be less useful to leaves than it seems. Here's the idea. Plants that absorb too much light, particularly in intense sunlight, can actually be damaged. Too many excited electrons, too much energy, flowing through the system at once, can overwhelm the machinery and cause harm. Green light, being reflected, acts as a kind of natural buffer. By not absorbing green light, the leaf avoids taking in so much energy that it damages itself on bright sunny days. This is still an area of active research, and not everyone agrees on the details, but it's a thought worth holding gently. The greenness of leaves might be in part a form of self-protection. Here's a quiet detour that's worth taking. Some scientists have proposed an idea, sometimes called the purple Earth hypothesis. The idea goes like this. The very earliest photosynthetic organisms, the first ones to use light for energy, might not have used chlorophyll. They might have used a different pigment called retinol. And retinol doesn't make things green, it makes things purple. If the earliest photosynthesizers used retinol, the ancient oceans and early land surfaces might have been covered in purple microbial mats, not green ones. And here's the interesting part. Retinol absorbs green light, the opposite of chlorophyll. So if the earliest ocean was carpeted in purple organisms, absorbing green light, there would have been a lot of green light bouncing back into the water, green light reflecting everywhere. When cyanobacteria evolved, using chlorophyll, absorbing red and blue light, they might have been able to thrive, precisely because they were using a part of the light spectrum that wasn't being used by the purple organisms. They were capturing the red and blue light that passed through the purple layer and used. It's a beautiful idea. The greenness of plants might be an evolutionary response to competition with even older purple photosynthesizers. The evidence for the purple Earth hypothesis is still somewhat speculative. It's a scientific proposal, not a settled fact, but it gives a sense of how rich and surprising the deep history of photosynthesis is. Life has been experimenting with light for billions of years. Let's go somewhere pleasant now. Imagine you're standing in a forest, not a dense jungle, just a quiet, temperate forest, the kind with tall trees, oaks or beeches or maples. It's midday in summer. Sunlight is coming down through the canopy. Look up. The light that reaches the forest floor is different from the light above the trees. The leaves above you have absorbed much of the red and blue light. They've taken what they need. What filters down through the canopy is a gentler, greener light. Forest light has a particular quality, a greenish softness. People who spend time in forests often find it calming, restful. There's something about that filtered light, the light that the trees have already processed, that feels gentle. This is actually one reason why spending time in forests is thought to be good for human well-being. There's a practice in Japan called Shinrin-yoku, forest bathing. It involves simply spending time in a forest, moving slowly and attentively. Many studies have found that this reduces stress hormones, lowers blood pressure, and improves mood. Part of this might be the light. The green filtered canopy light is genuinely different from the bright full-spectrum sunlight in an open field. It's quieter light, less stimulating. The forest floor is also a place of ecological competition. Plants that grow on the forest floor, ferns, wild garlic, mosses, have adapted to live in low light. They've evolved chloroplasts that are particularly efficient at capturing the limited light that reaches them. Some shade plants have chloroplasts that spread out flat, maximizing their surface area. When light is scarce, in bright light, those same chloroplasts may turn edge-on, reducing their surface area. To avoid absorbing too much, chloroplasts can move slowly, but they can reorient themselves inside the cell in response to light levels. This is called chloroplast movement, and it's another elegant, automatic mechanism that helps the leaf manage its light intake. We talked earlier about how there are more pigments in leaves than just chlorophyll. Let's spend some time with those other pigments. Carotenoids are a large family of pigments found in most plant leaves. They absorb blue and blue-green light and reflect yellow and orange wavelengths. But in most green leaves, the chlorophyll is so abundant that it masks the carotenoids completely. The yellow and orange is invisible beneath the dominant green. However, carotenoids do more than just contribute color. They also play a role In photosynthesis, carotenoids can absorb light and transfer that energy to chlorophyll. They act as accessory pigments, extending the range of light wavelengths that the leaf can use for photosynthesis. While chlorophyll is best at capturing red and blue light, carotenoids capture some of the blue-green light that chlorophyll misses and pass it along. It's a team effort. Chlorophyll is the main player, but the carotenoids help fill in the gaps. Carotenoids also have a protective role. Very high levels of light can damage photosynthetic machinery. Too many excited electrons, too much energy. Carotenoids help dissipate excess energy, converting it safely to heat, rather than allowing it to cause damage. They're a kind of safety valve. So the hidden yellow pigments in a green leaf are busy doing at least two things: helping to gather light and protecting the leaf from having too much of it. There are also anthocyanins, the pigments that make red, purple, and blue colors. In plants, in some leaves, anthocyanins are present alongside chlorophyll. The result can be striking: leaves that are dark red, or bronze, or purple, tinged with green. Think of certain ornamental plants with deep burgundy leaves, or young red leaves on some trees in spring. These red leaves contain both chlorophyll and anthocyanins. The anthocyanins give them their reddish color. What do anthocyanins do? This is still a question, with several possible answers. One idea is that anthocyanins protect young leaves from UV radiation. They absorb ultraviolet light that might otherwise damage the delicate, still developing leaf. Young leaves have very thin cuticles and are often more vulnerable to radiation. Another idea is that the red color deterrs insects. Some insects that prefer to lay their eggs on leaves may avoid red ones, perhaps because the color signals the presence of compounds that aren't suitable for their larvae. Both ideas have some supporting evidence. The true answer may involve a combination of factors. Now let's come to autumn. Autumn is when the hidden colors emerge, as days get shorter and temperatures drop. In the autumn, deciduous trees begin preparing to shed their leaves. This is not an accident or a failure. It's a deliberate, carefully orchestrated process called senescence. The tree begins to break down the chlorophyll in its leaves. It recycles the nitrogen from the chlorophyll back into the branches and trunk, saving it for the following spring. Chlorophyll is expensive to make. It contains nitrogen, which is often a limiting nutrient for plants. Rather than let it fall to the ground with the leaves and be lost, the tree reabsorbs it. As the chlorophyll breaks down, it fades, and as it fades, the other pigments, which were always there, just hidden, are revealed. The carotenoids, with their yellows and oranges, had been masked by green all summer. Now, with the green gone, they appear. A leaf that looked plain green all summer suddenly shows its gold, its amber, its warm orange. And then there are the brilliant reds and crimsons. Those often come from anthocyanins. But interestingly, in many trees, the anthocyanins are made fresh. In autumn, as the leaf is dying, they're not simply revealed, they're newly produced. Why would a dying leaf invest energy in making new pigment? There are several theories. One is that the anthocyanins help protect the leaf during the senescence process. As the leaf breaks down its own structures, it may be vulnerable to light damage, and the anthocyanins screen out harmful radiation. Another theory relates to insects. Some trees are attacked by aphids that overwinter on their bark. If the tree produces very vivid autumn colors, it might be signaling to the aphids that it is chemically defended, that it has lots of compounds. That would be unpleasant for the aphids. The aphids might then choose a different tree to overwinter on. Under this theory, autumn color is a kind of warning signal. This is called the coevolution hypothesis of autumn coloration, and while it's controversial and not universally accepted, it's a fascinating idea. Whatever the reason, for the reds, the result is the autumn landscape we know, the warm, glowing mixture of gold and amber and scarlet and deep red, all of it, the whole extraordinary display, is simply the hidden colors of leaves being revealed as the green fades away. For a brief time, the forest tells us what has been there all along. Let's step back in time again, not as far as before, but back a few 100 million years. The first land plants appeared about 470 million years ago. These early land plants were very simple, small, low-lying, without true leaves. They may have looked a bit like liverworts or hornworts, which still exist today in moist, shady places. Over tens of millions of years, land plants evolved to be taller and more complex. They developed roots to anchor themselves and absorb water. They developed conducting tissues, the ancestors of xylem and phloem, to move water and nutrients through their bodies, but they didn't have leaves. At first, the early versions of leaves probably evolved from branches that broadened and flattened over many generations. The first true leaf-like structures appear in the fossil record around 400 million years ago. During a period called the Devonian, these early leaves were small, simple things compared to the elaborate leaves of today's plants. But the basic idea was already there, a flat green structure for capturing sunlight. Over the following millions of years, leaves diversified enormously. They became larger, developed more complex venation patterns, developed specialized structures for different environments. Desert plants evolved small, thick leaves to minimize water loss. Tropical plants evolved enormous leaves to capture light in the shade beneath the forest canopy. Aquatic plants evolved floating leaves with waxy surfaces. Today, leaves come in an astonishing variety of sizes, shapes, and structures, but almost all of them share chlorophyll. Almost all of them are green. The greenness of leaves is one of the most ancient, most consistent features of plant life. It goes back to those first photosynthetic microbes in the ocean 2.5 to 3 billion years ago, through all the billions of years of evolution, through the incredible diversification of life on Earth. Chlorophyll has been the central molecule of photosynthesis. The green of a leaf is not a recent invention. It is among the oldest colors in the living world. Let's think about what photosynthesis has done to the Earth over billions of years. We mentioned earlier that the great oxygenation event, driven by cyanobacteria, transformed the Earth's atmosphere. But photosynthesis has continued to shape the atmosphere throughout the history of life on Earth. Today, photosynthesis by plants, algae, and cyanobacteria produces an enormous amount of oxygen, approximately 300 billion tons of oxygen per year. This is the oxygen that keeps the atmosphere at about 21% oxygen, the level that allows animals like us to breathe. If photosynthesis were to stop, if every plant and alga on Earth suddenly stopped producing oxygen, the oxygen in the atmosphere would gradually be depleted. It would take millions of years because there's so much oxygen already in the atmosphere, but it would happen. Every breath you take is sustained by the ongoing work of photosynthesis. Photosynthesis also affects carbon dioxide levels. Plants, algae, and cyanobacteria absorb enormous amounts of carbon dioxide from the atmosphere. They are, in the language of climate science, carbon sinks. They absorb more carbon than they release. The carbon in plants is ancient atmospheric. Carbon dioxide, when plants die and decompose, that carbon is released back into the atmosphere. But when plant material is buried and preserved over millions of years, as happened extensively during the Carboniferous period, about 300 to 360 million years ago, the carbon stays locked in the ground. The coal, oil, and natural gas that we burn today are the compressed remains of ancient plants and marine organisms that photosynthesized hundreds of millions of years ago. When we burn fossil fuels, we are releasing carbon dioxide that plants removed from the atmosphere long, long ago. In a sense, burning fossil fuels is like running photosynthesis in reverse, undoing hundreds of millions of years of carbon storage in a geological instant. This is one reason why atmospheric carbon dioxide levels have been rising and why this matters for the climate. But that is a large topic for another time. Let's go back to the chlorophyll molecule one more time and look at it a little more carefully. We said that chlorophyll has a porphyrin ring structure with a magnesium atom at its center. Let's think about why magnesium. Magnesium is a metal, element number 12, on the periodic table. In its pure form, it's a silvery white solid, but when magnesium atoms sit at the center of a porphyrin ring, they allow the ring to engage with light in a very specific way. The electrons in the porphyrin ring are not tightly fixed in one place. They're somewhat delocalized. They can spread across the whole flat ring structure. This delocalization is what makes chlorophyll so effective at absorbing light. When a photon of the right wavelength hits the chlorophyll molecule, it interacts with these delocalized electrons. One of the electrons absorbs the photon's energy and jumps to a higher energy level. The exact energy of that jump corresponds to the specific wavelengths of red and blue light. If the magnesium were replaced by a different metal, the energy levels would shift. Different wavelengths of light would be absorbed. The molecule would have a different color. Hemoglobin, the molecule that makes blood red, has a very similar porphyrin structure, but at the center of hemoglobin's porphyrin ring is iron, not magnesium. Iron changes the energy levels of the electrons, and so hemoglobin interacts with light differently. It absorbs blue and green light. It reflects red. That's why blood is red. Chlorophyll has magnesium. Hemoglobin has iron. The color of a leaf and the color of blood both come down to which metal sits at the center of a porphyrin ring. It's a quietly wonderful connection. The green of a leaf and the red of blood are variations on the same theme, the same basic molecular structure, with one atom swapped, produces two of the most iconic colors in the living world. Here's a gentle forward-looking thought. Scientists have long been interested in the possibility of artificial photosynthesis, creating man-made systems that do what leaves do, systems that capture sunlight and use it to produce fuel or other useful chemicals. The appeal is obvious. Sunlight is abundant and free. Carbon dioxide is in the atmosphere in large quantities. If we could build artificial leaves, machines that convert sunlight and carbon dioxide into usable fuel, we would have a potentially unlimited, clean energy source. Researchers have made real progress on this. There are now experimental devices that use sunlight to split water, just as photosynthesis does, producing hydrogen gas, which can be used as a fuel. Other experimental systems use sunlight to convert carbon dioxide into simple organic molecules. None of these artificial systems are as efficient or as elegant as a real leaf, yet evolution has had billions of years to refine chlorophyll, and the photosynthetic machinery human engineers have had perhaps a century. The gap is still large, but the gap is narrowing, and there's something fitting about it that after billions of years of plants quietly capturing sunlight in the green machinery of their leaves, we are now trying to understand and replicate that machinery to learn from the leaf. The greenness of leaves has sustained life on Earth for billions of years. It might help sustain it for billions more. We've been talking mostly about the kind of photosynthesis done by land plants, what's called oxygenic photosynthesis. Oxygenic because it produces oxygen as a byproduct, but not all photosynthesis is like this. Some bacteria do a different kind of photosynthesis. These are called anoxygenic photosynthesizers. They photosynthesize without producing oxygen. Instead of splitting water, they split other molecules. Some use hydrogen sulfide, some use hydrogen gas, some use iron compounds. These bacteria tend to live in unusual environments, deep in the ocean, in hot springs, in sulfur-rich environments. They don't look green. Some are purple, some are red, some are brown. Their photosynthetic pigments are different from chlorophyll. They absorb different wavelengths of light. They've adapted to use whatever light reaches their unusual habitats. The diversity of photosynthetic strategies in the microbial world is remarkable. Light is such a valuable energy source that evolution has found many different ways to harness it, using many different molecules, absorbing many different wavelengths. The green version, chlorophyll-based, oxygenic photosynthesis, is the most ecologically dominant on Earth today. It's the one that shaped the atmosphere and made complex life possible. But it's just one approach among many. Let's think about the relationship between leaves and animals for a little while. Almost all of the energy in the food chains of terrestrial ecosystems, the food chains on land, comes ultimately from photosynthesis, from leaves, or from other photosynthetic organisms. A caterpillar eats leaves. The caterpillar is eaten by a bird. The bird is eaten by a hawk. Every step up that chain, energy is lost. Most of the energy in each organism's food is used up just to keep it alive, and only a fraction passes on to the next predator. But all of that energy, at every step, came originally from sunlight, captured by leaves, stored in sugar, passed along through the food chain. Step by step, you eat food. That food, whether it's a vegetable or a piece of meat, contains energy that came from plants. Even meat from an animal that ate only other animals can trace its energy back through enough steps to photosynthesis. The energy in your body right now, the warmth that keeps you comfortable, the activity of your brain as it processes these words, the gentle rise and fall of your breathing, all of that energy came from sunlight, captured in the green machinery of leaves. In a very real sense, you are powered by photosynthesis. You are made, in large part, of carbon that was once carbon dioxide in the air, fixed into organic molecules by chlorophyll, passed along through the food chain until it reached you. Your carbon and the carbon in a leaf have a common story. Let's rest for a while. In the cultural world, human beings have always lived among leaves. We evolved in landscapes full of plants. Our relationship with the green world is ancient and deep. Leaves appear throughout human art and architecture. The acanthus leaf, with its deeply cut lobes and curling edges, inspired a decorative motif used by ancient Greeks and Romans. It appears on the capitals of Corinthian columns. the carved stone tops of those elegant Greek pillars. It spread through Roman architecture and then into Byzantine, Renaissance, and beyond. The influence of a single leaf shape on human design has stretched across thousands of years. In Japan, the maple leaf, the momiji, is a symbol of autumn and impermanence. The tradition of viewing autumn leaves, koyo, is as culturally significant as the cherry blossom viewing. In spring, people travel to see the red and gold of maples in the mountains. Poets have written about it for centuries. In Celtic traditions, the oak leaf and the four-leaf clover have deep symbolic importance. The oak was sacred, associated with strength, endurance, and wisdom. A grove of ancient oak trees was a sacred place in many early European cultures. In Hinduism and Buddhism, the lotus leaf has special significance. The lotus grows in muddy water, but produces pristine white flowers and perfectly clean leaves. The lotus leaf is famously waterproof. Water beads up on its surface and rolls off, carrying dirt with it. This property is called the lotus effect, and it has inspired water-repelling materials. In modern engineering, the lotus leaf, as a symbol, represents purity, the ability to remain clean and untouched, even in difficult circumstances. Leaves are everywhere in human symbolism, because leaves are everywhere in human life. We are a species that evolved, surrounded by leaves. The green world shaped us. It fed us, clothed us, sheltered us, provided us with the air we breathe. It's no wonder that leaves have found their way into our deepest stories and symbols. There is a kind of language in leaves, not a language that speaks in words, but a language of information, signals that leaves send out, and signals that other organisms receive. When a plant is being eaten by a caterpillar, for example, the damaged leaf can release chemicals into the air. These chemicals are sometimes called volatile organic compounds. They drift through the air and can be detected by neighboring plants. Some nearby plants, receiving these chemical signals, respond by ramping up their production of defensive compounds in their own leaves, making themselves less tasty before they've even been attacked. This is sometimes called plant communication. It's a controversial term. Plants don't communicate the way animals do, but the information transfer is real. Damaged plants release compounds. Nearby plants respond. Something is being transmitted. There's also communication. Underground, plants are connected by networks of fungi, mycorrhizal networks, that extend through the soil and connect the roots of many different plants. Through these fungal networks, carbon, nutrients, and perhaps chemical signals can move from one plant to another. A large tree can transfer carbon to smaller, shaded seedlings through the fungal network. In a healthy forest, the trees are not simply competing with each other. They are, in a limited but real sense, connected, supporting each other. Some scientists call these networks the Wood Wide Web. It's a catchy name for something genuinely fascinating, though the extent and significance of plant-to-plant communication through fungal networks is still being actively researched and debated. But the basic fact stands: the green world is not passive and silent. Leaves exchange information with the world around them, through the air and through the soil. They're busy and connected and quietly responsive to their environment. You may be getting close to sleep now. That's good. That's what we're here for. Let's imagine something together. You're walking through a garden at night. It's warm, but not too warm. There's a soft breeze. The moon is out, and it's nearly full, so there's a gentle, silver light falling over everything. The leaves around you are dark. In the moonlight, the bright green of the daytime is softened into deep, dark tones, dark green, nearly black, in places with silver at the edges where the moonlight catches them. The leaves are resting now. Photosynthesis has stopped. For the night, there's no sunlight, so there's no energy to capture. The chloroplasts are quiet. The stomata are mostly closed. The leaf is conserving water, resting, waiting for morning. But even in the dark, the leaf is still alive. The cells are still breathing, doing what's called cellular respiration, the opposite process from photosynthesis. They're slowly breaking down some of the sugar made during the day, releasing a small amount of carbon dioxide, using the energy to maintain themselves very slowly, very gently, just enough to stay alive through the night. Tomorrow morning, when light first appears, the stomata will begin to open. The chloroplasts will come back to life. The electrons will begin to jump again. The Calvin cycle will begin turning. Oxygen will begin to flow. But for now, the garden is still and quiet, and you are almost asleep. If you're still with us, let's go a little deeper. Let's explore the chlorophyll molecule. One more time, we've talked about the porphyrin ring and the magnesium at its center. We've talked about the phytol tail, but there's something else worth mentioning. Chlorophyll molecules don't work alone. Inside the chloroplast, they work in groups, clusters of chlorophyll and related molecules, perhaps 200 to 400 in each cluster, form what's called an antenna complex. The antenna complex is like a light harvesting network. Each chlorophyll molecule in the cluster can absorb a photon of light, but rather than immediately using that energy, the excited electron passes its energy to a neighboring chlorophyll molecule, which passes it to another and another until it reaches a special pair of chlorophyll molecules at the center of the complex. This central pair is called the reaction center. The reaction center is where the real photochemistry happens. When energy arrives from the antenna complex, the reaction center uses it to drive an electron transfer, the first step in the electron transport chain that eventually produces ADP and splits water. The antenna complex is an extraordinarily efficient light-capturing device because there are hundreds of chlorophyll molecules working together. The complex can capture a much wider range of light than any single molecule could. Energy hops from molecule to molecule incredibly rapidly in a matter of femtoseconds. A femtosecond is 1/400th of a second. The energy transfer is essentially instantaneous. The efficiency of this process is remarkable. Quantum mechanical calculations have shown that the energy transfer in photosynthesis approaches the theoretical maximum efficiency allowed by physics. Plants have evolved a system of light harvesting that is nearly as good as it is physically possible to be. This is not an accident. Billions of years of natural selection have refined the antenna complex and the reaction center into an extraordinarily precise, extraordinarily efficient machine, all inside a structure too small to see, inside a chloroplast, inside a cell, inside a leaf, inside the green, outside your window. There's a very quiet, very interesting field of science that has emerged in recent years. It's called quantum biology. Quantum mechanics is the branch of physics that describes the behavior of matter at very small scales, the scale of atoms and molecules. At this scale, matter behaves in ways that are strange and counterintuitive. Particles can exist in multiple states at once. They can tunnel through barriers. Energy can be transferred in ways that classical physics doesn't predict. For a long time, scientists thought that quantum mechanical effects were only important in the cold, isolated conditions of a laboratory. The warm, wet, noisy environment of a living cell seemed too chaotic for delicate quantum effects to survive. But in 2007, researchers studying photosynthesis found something surprising when they illuminated molecules in a photosynthetic complex with carefully tuned laser pulses. They observed patterns in the emitted light that could only be explained by quantum coherence. The energy transfer between chlorophyll molecules appeared to be happening in a quantum mechanical way, multiple pathways being explored simultaneously, as if the energy were in multiple places at once. This was startling. Quantum coherence in a warm, wet, biological system at room temperature. The interpretation and significance of these findings has been debated extensively in the scientific community. Some researchers believe that quantum effects genuinely improve the efficiency of energy transfer in photosynthesis. That evolution has found a way to harness quantum mechanics to make light harvesting more efficient. Others are more skeptical, arguing that the quantum effects observed are incidental rather than functional. The debate continues. The science is active and evolving, but it raises the possibility that a leaf, an ordinary green leaf on an ordinary tree, might be performing quantum mechanical computation as it captures sunlight. That at the most fundamental level, a leaf's greenness connects to the deep strangeness of quantum physics. It's a thought to hold very lightly, very gently, in the way you might hold a delicate dream. Let's wander for a while through the incredible diversity of leaf forms. A leaf can be almost anything. The giant water lily, Victoria amazonica, produces leaves that can be 2 meters across, large enough for a small child to stand on. These enormous, floating platforms have raised edges and a ribbed underside structure that distributes weight. They're engineering marvels, growing in the still waters of Amazonian lakes. At the other extreme, some desert plants have leaves reduced to tiny scales, almost invisible. In these plants, the green photosynthetic work is done by the stem. The whole trunk, or branch, takes over the role of the leaf and is green and photosynthetically active. Cacti are the most familiar example of this. The flat, paddle-like structures of a cactus are modified stems, not leaves. The cactus's true leaves are the spines, highly modified, reduced leaves that have lost their photosynthetic function entirely and serve purely as defense. So the cactus spine and the maple leaf are, in the grand scheme of plant evolution, variations on the same structure, one reduced to a hard, sharp defense, one broadened into a wide, light-harvesting surface. Carnivorous plants take this variation to its extreme. The pitcher plant has a leaf modified into a deep, fluid-filled cup, a trap for insects and other small animals. The Venus flytrap has leaves modified into hinged snap traps with sensitive trigger hairs that detect the presence of an insect. These are still leaves. In the evolutionary sense, they're still made from the same tissues, with the same basic cellular structure. They've just been shaped by evolution, into something quite different. And often, even in these modified leaves, there is still chlorophyll. The pitcher plant is still green. It still photosynthesizes, still captures sunlight, even as it also captures flies. The oldest fossil leaves we've found are ancient, but leaves themselves have an even longer photosynthetic history. The earliest structures we might reasonably call leaves in the fossil record are about 400 to 380 million years old. From the Devonian period, these early leaves were found on early vascular plants, plants with proper conducting tissues. But before true leaves, photosynthesis in plants was carried out by flat green, leaf-like thalli, the flat bodies of liverworts and hornworts, and before that, cyanobacteria in the ocean were photosynthesizing for over 1 billion years before the first land plants appeared. The continuity is remarkable. The same basic photosynthetic process, using the same basic molecule, chlorophyll, has been the foundation of almost all life on Earth for over 2.5 billion years. The leaf outside your window is not just a leaf, it is the latest expression of a lineage that stretches back almost to the dawn of complex life on Earth. It carries within it the accumulated refinements of billions of years of evolution. The green in a leaf is very old. Green. There is mathematics. In leaves, many leaves arrange themselves on their stems according to mathematical patterns called phyllotaxis. Phyllotaxis comes from the Greek for leaf arrangement, and the arrangements that plants use, when looked at carefully, often follow Fibonacci numbers. Fibonacci numbers are the sequence 1, 1, 2, 3, 5, 8, 13, 21, 34, 55, 89. Each number is the sum of the two before it. If you look at the spiraling arrangement of leaves on a stem, or the scales of a pine cone, or the seeds in a sunflower head, you will often find that the spirals come in Fibonacci numbers. A sunflower head might have 34 spirals going one way and 55 going the other. A pine cone might have 8 spirals in one direction and 13 in the other. Why Fibonacci? The explanation involves the mathematics of packing. New leaves on a stem are produced from a growing tip, one at a time. Each new leaf is positioned at a certain angle relative to the previous one. The angle that minimizes overlap between leaves, that ensures each leaf gets as much light as possible without blocking others, turns out to be very close to the golden angle, which is approximately 137.5 degrees. And when you tile a spiral using the golden angle, the number of visible spirals you get are always consecutive Fibonacci numbers. So the Fibonacci patterns in leaves and cones and flowers are not a coincidence or a mystery. They are the natural, geometric consequence of arranging structures around a growing point using the golden angle. And the golden angle is optimal because it maximizes light exposure. The mathematics of leaves is the mathematics of efficiency, of finding the arrangement that allows maximum sunlight to reach every leaf, all without any planning or design, just geometry, growth, and the quiet pressure of natural selection. It's getting later now. The world outside is quiet. The leaves out there are quiet too. Photosynthesis stopped hours ago. The chloroplasts are resting. The stomata are mostly closed. The whole plant is in a kind of metabolic nighttime mode. Think about all the things we've traced together tonight. We started with the simple question, why are leaves green? And we found that the answer leads through chemistry to the chlorophyll molecule with its porphyrin ring and its central magnesium atom, through physics to the nature of light, to electrons jumping between energy levels, to quantum mechanics, through evolutionary history, to cyanobacteria 3 billion years ago, to the endosymbiotic event that created chloroplasts, to the Devonian forests of 380 million years ago. The greenness of a leaf connects to the origin of oxygen in our atmosphere, to the food chains that support all complex life, to the ancient fossil fuels encoded in the earth, to the air you're breathing right now. A leaf is so ordinary and so extraordinary, both at once. Let's linger a little more on the inside of the leaf. There's something calming about going deeper and deeper into a small, quiet world. We mentioned the palisade mesophyll and the spongy mesophyll earlier. Let's spend a little more time there. The word mesophyll means middle leaf. It's the tissue in the interior of the leaf, between the upper and lower epidermis. This is where most of the chloroplasts live. In the palisade mesophyll layer, the cells are elongated and tightly packed, like a fence of columns standing shoulder to shoulder. Most of the chloroplasts in a leaf are concentrated in these cells. They're positioned to catch incoming sunlight efficiently. A palisade cell can contain 50 to 100 chloroplasts, and the chloroplasts within the cell can move, slowly rotating and reorienting, to catch more or less light, depending on conditions. In bright light, chloroplasts tend to orient themselves, with their flat faces parallel to the cell wall, edge on to the incoming light, reducing their absorption surface and preventing overload. In dim light, the chloroplasts rotate to face the light directly, maximizing their absorption surface. This slow, silent dance of the chloroplasts inside the leaf cells is happening constantly, all day in every leaf. The chloroplasts are adjusting their position in response to light. Below the palisade layer, the spongy mesophyll has more loosely arranged cells, with large air spaces between them. Carbon dioxide diffuses through these air spaces, reaching all of the cells in the leaf interior. The air spaces connect to the outside. Through the stomata, you can think of the leaf interior as a kind of soft green sponge, threaded through with veins and riddled with air passages, with the palisade layer on top, catching the light, and the spongy layer below, managing the gases, all of it working together, quietly. Let's imagine the moment of dawn. The sky is lightening, the stars are fading, there's a pale band of light on the eastern horizon. On a leaf, the first photons of sunlight are beginning to arrive, very gentle at first, a few thousand photons a second, an almost negligible flow of light energy by the standards of midday, but the chloroplasts respond immediately. Guard cells begin to absorb water, beginning their slow inflation. Stomata begin to open. Carbon dioxide begins to diffuse in. The first excited electrons jump upward in the reaction centers, very slowly, very gently, like waking up. The leaf's photosynthetic rate increases gradually through the morning. As light intensifies, around mid-morning on a sunny day, the leaf is operating at close to full capacity. Oxygen is pouring out of the stomata. Sugars are flowing down the phloem toward the rest of the plant. By midday, if it's very hot and sunny, the leaf may actually partially close its stomata to prevent too much water loss. It throttles back photosynthesis slightly to conserve water. It's a trade-off, a compromise between making sugar and staying hydrated. In the afternoon, as the light angle shifts and the temperature begins to drop, the leaf adjusts again, always adjusting, always responsive. And as evening comes, the light falls below the threshold for photosynthesis. The stomata close, the chloroplasts stop working. The leaf settles into its nighttime chemistry. Every day, This cycle, every single day of the growing season, the leaf waking, working, resting, just like you. Let's venture into the water. For a little while, the ocean is green too. In many places, algae, from the microscopic phytoplankton to the great kelp forests, are among the most important photosynthetic organisms on Earth. Estimates suggest that marine algae and phytoplankton are responsible for roughly half of all the photosynthesis that occurs on Earth, half of the world's oxygen production, half of the carbon fixed into organic molecules each year. The open ocean, from the surface, may look blue, but in those upper layers of sunlit water, invisible to the naked eye, trillions of phytoplankton cells are photosynthesizing, each one tiny, but together they constitute one of the most important ecological forces on the planet. Many phytoplankton contain chlorophyll, the same basic molecule as in land plants. They too are green, or greenish, but many also contain other photosynthetic pigments that give them different colors. Some are golden brown, some are blue-green, some are red. In the ocean, light penetrates to different depths. At the surface, there's full-spectrum sunlight. At greater depths, red light is quickly absorbed by the water, and only blue-green light penetrates. Photosynthetic organisms that live deeper in the water column have evolved pigments that absorb blue-green light efficiently, rather than the red-absorbing chlorophyll that works best in surface light. Red algae, the seaweeds, that often appear deep red or purplish, contain pigments called phycobiliproteins that absorb blue-green light. In the deep waters where they often live, this is exactly the kind of light available. The color of a photosynthetic organism is a window into where it lives and what kind of light it has access to. Green at the surface, red and brown in the deeper water. Life adapted to its light environment, as it always has. We've spent a lot of time with green, but let's take a brief, gentle tour through the other colors of photosynthesis in the living world. The red of red algae, phycobiliproteins absorbing blue-green light. In deeper water, the golden brown of diatoms and many phytoplankton, a pigment called fucoxanthin, absorbing blue-green light. The blue-green of cyanobacteria, a mixture of chlorophyll and phycobiliproteins. The purple of purple bacteria, bacteria chlorophyll, absorbing near-infrared light. And the green of plants, chlorophyll A and B, absorbing red and blue. Each of these organisms has evolved a slightly different version of the same basic trick, capturing the energy of light and converting it to chemical energy. Each has adapted its pigments to the light available in its particular habitat, and together, all of these photosynthetic organisms, with all of their different colors, perform the global work of photosynthesis. They maintain the oxygen in our atmosphere. They remove carbon dioxide. They build the base of nearly every food chain on Earth. The diversity of colors in the photosynthetic world is a record of evolutionary experimentation, of billions of years, of life finding new ways to capture the light. Let's move away from science for a moment and into the sensory world of leaves. You know, the smell of rain on dry leaves and earth. There's even a word for it, petrichor. It comes from the Greek petra, stone, and ichor, the fluid that flows in the veins of the gods. In Greek mythology, petrichor is the smell of rain on dry ground. Part of this smell comes from the earth, from a compound called geosmin, produced by soil bacteria, but part of it comes from the oils and compounds that accumulate on dry leaves and soil during dry periods, which are released when rain hits. It's one of the most universally loved smells. Studies across many cultures have found that the smell of rain on earth and leaves is overwhelmingly pleasant. To humans, perhaps because it signals water, and water means green, and green means food, and life. The smell of a green leaf itself, the fresh, clean smell, when you crush a leaf between your fingers, comes from compounds called green leaf volatiles. These are released when leaf cells are damaged. They include compounds with scientific names like hexanol and hexanol, but you know them simply as the smell of fresh grass, of torn leaves, of summer. These compounds are sometimes released when an animal eats a plant. They serve as signals, warning neighboring plants or attracting predators of the animals that are eating the leaf. So the lovely smell of fresh grass being cut is in part a distress signal from the grass. It's a little bittersweet, but the smell is still beautiful, and the leaf is still green. Let's think about winter. In temperate climates, deciduous trees lose all their leaves. In autumn, the trees stand bare through the winter months, skeleton branches against gray sky, no green anywhere. From the outside, they look dead, but they're not dead. They're dormant, deep inside the bark. In the cambium, the thin layer of dividing cells just under the bark, the tree is alive, maintaining itself, waiting. The roots are still alive, in the soil, slowly absorbing whatever water is available. In spring, when days lengthen and temperatures rise, the tree wakes. New buds that were set in autumn begin to swell. The folded embryonic leaves inside the buds begin to expand, and within days, the tree is covered in a pale, fresh, extraordinarily delicate green. The first leaves of spring are a particular kind of green, soft, pale, almost luminous. They're thinner than summer leaves, with less chlorophyll, letting more light through. They tremble in the lightest breeze. This fresh spring green is one of the most beloved colors in the temperate landscape. There are Japanese words for it, the particular green of new spring leaves, the quality of light through fresh leaves. It's celebrated in poetry, in painting, in music. It's just chlorophyll, just electrons, absorbing photons, just physics and chemistry. But it's also somehow among the most beautiful things in the world. Chlorophyll itself, extracted from plants, has been used by humans for various purposes. As a food coloring, chlorophyll gives a green color to certain products. It's used in some candies, drinks, and condiments. It's also added to certain health foods and supplements. Chlorophyll and its derivatives have been investigated for various health properties. Chlorophyllin, a water-soluble, semi-synthetic derivative of chlorophyll, has been studied for potential antioxidant and detoxifying properties. In medicine, a chlorophyll-derived compound is used in a treatment called photodynamic therapy. Certain porphyrin-based molecules related to the porphyrin ring of chlorophyll can be delivered to cancer cells. When these molecules are then activated by light, they produce reactive oxygen species that damage the cancer cells. It's a remarkable twist. The same basic molecular structure that captures light energy in a leaf can be adapted to destroy cancer cells when illuminated. The porphyrin ring, which has been at the center of photosynthesis for billions of years, finds new work in human medicine. Let's slow down even more. You've been with us for a long time now. We've traveled through chemistry, history, biology, physics, and poetry. We've gone inside the molecule and out into the history of the planet. We've walked through forests and dove into oceans, all because of a simple question: why are leaves green? The answer, in its simplest form, is because chlorophyll absorbs red and blue light and reflects green light back to our eyes. But you know now that this simple answer rests on an extraordinary foundation. on the structure of a molecule, on the quantum behavior of electrons, on evolutionary history, spanning billions of years, on the chemistry of the early Earth's atmosphere, on ancient partnerships between cells. The greenness of leaves is one of the most fundamental features of life on Earth. It is older than animals, older than complex cells, older than the oxygen in our atmosphere, older than almost anything we can point to in the living world. And yet, it is present right now, in every garden, every park, every field, every forest, every meadow, ordinary and extraordinary at the same time. As we move toward the end of our time together, let's follow the chloroplast one last time through its day. In the morning, the chloroplast is activating. The enzymes of the Calvin cycle are being switched on. The thylakoid membranes are ready, their chlorophyll molecules poised. Through the day, electrons jump and fall. Water is split. Oxygen drifts out. Carbon dioxide drifts in. ADP is made and used. Sugars accumulate. Through the afternoon, as light slowly fades, the rate of photosynthesis decreases. The Calvin cycle slows. As evening comes, the light falls below the compensation point, the level at which photosynthesis and respiration are exactly balanced. Below this, the plant respires more than it photosynthesizes. Carbon dioxide begins to come out of the leaf rather than going in. And then, the night, the chloroplast is quiet. Still, the thylakoids float. In the stroma, the chlorophyll molecules are dark. No electrons jumping, no photons absorbed. The whole system waits, patient, for morning. It has done its work today. Sugar has been made. The plant has grown, perhaps by a tiny amount. The branch is perhaps a fraction of a millimeter longer. A leaf bud somewhere is perhaps imperceptibly fuller. The tree is very slightly larger than it was yesterday. From sunlight, from air, from green. Let's end with one final, gentle, wide thought. Sunlight is the energy source for nearly all photosynthesis. On Earth, the sun is the star our planet orbits. It's a middle-aged, medium-sized star. About halfway through its life, it formed about 4.6 billion years ago from a cloud of gas and dust. The sun will last for another 5 billion years or so, long enough, from our perspective, to be effectively eternal. But there are other stars, billions of them, in our galaxy alone, trillions, across the observable universe. Many of these stars have planets, and some of those planets orbit in the habitable zone. The range of distances from a star where liquid water could exist, on the surface, where life, as we understand it, might be possible. If there is life elsewhere in the universe, and if that life evolved on a planet with a star to orbit, it might have evolved photosynthesis. It might have evolved a way to capture light energy and store it in chemical bonds. It might have evolved pigments that absorb some wavelengths and reflect others. The reflected light would give it a color, and that color might not be green. It might be any color, depending on the spectrum of the host star. Life on a planet orbiting a reddish star might use pigments that absorb the prevalent wavelengths of that red light, pigments that might appear purple or black. Life on a planet orbiting a very hot blue star might use pigments that appear yellow or orange. The universe might be full of photosynthesis, full of living things, capturing their star's light to make food, and they might be every color imaginable. But here on Earth, around our particular yellow sun, the color evolution settled on is green, the green of chlorophyll, the green of the world outside your window, the green that came from cyanobacteria in an ancient ocean, carried forward by billions of years of evolution into every leaf of every plant on this particular blue planet. We are coming to the end now. It's been a long, gentle journey through chemistry and biology and history and earth science and physics and culture and poetry, all stemming from the green in a leaf. You've done very well. You don't need to remember any of this. It's just something that drifted through you on a quiet night. The leaves outside are dark now. The chloroplasts are resting. The stomata are closed. The sugars made today are moving through the plant, nourishing it, building it, strengthening it. For tomorrow, the whole green world is asleep, or very nearly, and perhaps you are too. Just let your body settle. Let the thoughts drift. The electrons and the photons, the porphyrin rings, the guard cells, opening and closing, the ancient cyanobacteria, in their ocean, the first leaves of the Devonian, the hidden yellows of autumn, the smell of petrichor, the mathematics of Fibonacci, the quantum dance in the antenna complex. Let all of it drift away gently, like a leaf on still water. You know now why leaves are green, or rather, you know the beginning of the answer, because the truth is, the more closely you look at anything in nature, any single ordinary thing, the deeper and wider and more surprising it gets. A leaf is not simple. It is not just a flat green thing on a twig. It is a portal into the history of life, the physics of light, the chemistry of molecules, the ancient story of a planet that turned green. But tonight, that's enough. That's more than enough. The leaf is green. The world is quiet, and you can rest now. If you're still awake, just barely, here is one last thought to carry you into sleep. Right now, somewhere in the world, it is dawn, in a forest somewhere, perhaps in a mountain range, in Japan, or on the slopes of the Appalachians, or in the forests of the Andes, or in the vast boreal forests of Canada. The first light of morning is touching the first leaves. The guard cells are just beginning to swell. The first electron somewhere is just being lifted by the energy of the first photon of the day. A new cycle of photosynthesis is beginning, a quiet, ancient, continuous process, the same process that has been happening every morning for billions of years, the same process that first filled our atmosphere with oxygen, the same process that built the world we live in. Just one electron, just one photon, just one leaf, but multiplied by trillions of leaves across the whole sunlit earth. It is the engine of all life, and it is green. Sleep well. For those of you who are still gently with us, still drifting on the edge of sleep, let's wander through everything we've talked about one more time. Very slowly, very gently, like reading the same poem a second time, slower than the first. A leaf is a solar panel. It captures sunlight and uses it to make sugar. This process is called photosynthesis, and it happens inside tiny structures called chloroplasts, inside the cells of the leaf. Inside the chloroplast, there is a molecule called chlorophyll. Chlorophyll is a pigment. It absorbs some colors of light and reflects others. Specifically, chlorophyll absorbs red and blue light very efficiently. It does not absorb green light very well, so green light bounces back. That's the color we see. The green of a leaf is the light the leaf didn't need. Chlorophyll has a flat, ring-shaped core called a porphyrin ring. In the center of this ring sits a single magnesium atom. This specific arrangement of atoms is what makes chlorophyll absorb the wavelengths it does. Different atoms in the center would absorb different wavelengths and produce different colors. Hemoglobin, the molecule that makes blood red, uses iron instead of magnesium, in a very similar porphyrin structure. When a photon of red or blue light hits a chlorophyll molecule, an electron absorbs its energy and jumps to a higher energy level. This excited electron is then passed along a chain of molecules, the electron transport chain, that uses its energy to make ADP, the universal energy currency of life, and to split water molecules. When water is split, oxygen is released. That oxygen is what we breathe. Photosynthesis also runs a second cycle, the Calvin cycle, that uses the energy from ADP to take carbon dioxide from the air and build sugar. The carbon in the sugar came from the air. Most of a plant's body, most of its mass, is made from carbon dioxide from air. The chloroplast is not a random structure. It evolved from a free-living bacterium, a cyanobacterium, that was engulfed by a larger cell about 2 billion years ago and never left. Over time, it became permanently incorporated into the cell as an organelle. This is called the endosymbiotic theory, and it's one of the most elegant and well-supported ideas in biology. There are other pigments in leaves besides chlorophyll. Carotenoids are yellow and orange pigments that are always present, but usually hidden by the more abundant chlorophyll. In autumn, when chlorophyll breaks down, the carotenoids are revealed, the reds and golds of autumn foliage. Some red autumn colors come from fresh anthocyanins, produced as the leaf ages. The structure of the leaf supports all of this. The palisade mesophyll layer, just below the upper surface, is packed with chloroplasts for light capture. The spongy mesophyll below has air spaces for gas exchange. Veins carry water in and sugar out, stomata, tiny pores on the leaf's surface, guarded by pairs of curved cells, allow carbon dioxide in and oxygen and water vapor out. Photosynthesis began with cyanobacteria in the ancient ocean, perhaps 2.5 billion years ago. They slowly produced the oxygen that fills our atmosphere today, a process called the Great Oxygenation Event. This changed the Earth forever, eventually making complex life possible. The first true leaf-like structures appeared in the fossil record about 400 million years ago. Since then, leaves have diversified into an extraordinary range of shapes and sizes, from the 2-meter water lily to the microscopic scales of some desert plants. But almost all of them are green. Almost all of them contain chlorophyll. The greenness of leaves is woven into the history of life on Earth. It is older than animals, older than complex cells, nearly as old as life itself. And it is present, right now, in every plant in the world, in every quiet garden, in every sleeping forest, in the houseplant by the window, in the moss on the wall. The green of leaves is ancient and ordinary and extraordinary, all at once. And now it's time to rest. The leaf is quiet. The chloroplast is still. The electron has settled. Sleep.